Hi everyone, this is 8th video in our Advanced Pharmaceutical Dissolution Science and Application series. In this video, we will discuss two-stage and multi-stage dissolution testing and its applications. This is essential for enteric-coated tablets, delayed release, modified release systems, and product that requires GI simulation. Also, for many modern dosage forms, a single-stage dissolution test is not enough. Profile comparison is key to understanding real drug release behavior. A single medium dissolution test cannot capture formulation transitions like acid resistance or intestinal activation. The reason is simple. Drug release changes as the environment changes. That's why multi-stage dissolution testing is used to mimic stomach and intestinal condition and reflect real in vivo behavior. The 2-hour exposure in acidic media is not arbitrary. It aligns with gastric residence time recommendations in USP and FDA guidance. This reflects how long a dosage form typically remains in the stomach before moving to the intestine. Two-stage dissolution testing is the simplest and most common approach. The product is first exposed to an acidic medium, then shifted to the higher pH buffer to simulate intestinal conditions. This setup helps capture realistic environment-driven drug release behavior. Acceptance criteria are defined either in compendial monograph or during method development, depending on the product. For enteric-coated tablets, testing can be done by media replacement or by buffering the same vessel to shift pH. Both approaches are acceptable when scientifically justified. This is how enteric-coated products are evaluated. First, confirm acid resistance and then ensure rapid and complete release in intestinal condition in line with USP and FDA expectation. That's how real GI performance is confirmed in vitro. There are two accepted ways to create a pH transition in dissolution testing. For products sensitive to ionic strength or osmolarity, media replacement is preferred to avoid sudden compositional changes. Alternatively, pH can be shifted by adding a concentrated buffer to the same vessel. Both approaches are acceptable. The key is choosing the method that best reflects product behavior and is scientifically justified. Colon-targeted dosage forms often requires multi-stage dissolution testing. For example, pH 1.2 to pH 6.8 to pH 7.4 with extended exposure time. This approach is used when a product designed to release drug in multiple phases as it moves through GI tract. Multi-stage testing ensures the formulation performs exactly where and when it is intended to release. In multi-stage dissolution testing, we aim to mimic human GI transit from the stomach to the duodenum, then to the jejunum, and finally ileum. For modified release products, this means exposing the dosage form to multiple pH stages to evaluate how the matrix or coating behave across the intestinal tract. This approach helps confirm that the drug is released at the right place and at the right time. These approaches are widely used for BCS class 2 drugs and IVIVC modeling. Biorelevant media provide a more realistic prediction of in vivo behavior, especially for poorly soluble drugs. They are also critical for understanding food effects, where standard media may fail to reflect real GI conditions. It's critical to document the scientific rationale during method development. For delayed release and modified release products, regulators expect pH transition testing. This is both scientific necessity and a regulatory requirement. Clear documentation ensures the method is defensible, reproducible, and inspection ready. In day-to-day lab work, multi-state dissolution testing can be challenging. Common issues include pH drift, timing error, media handling, and variability. That's why method robustness is critical. The good news is that most of these challenges can be fixed with simple laboratory adjustment when the method is well designed and well controlled. In multi-stage dissolution testing, early drug release in acidic media usually indicates coating failure. On the other hand, rapid release in buffer confirms the formulation is functioning as intended. Interpretation focuses on one key question: does each stage performs as expected? That means no premature release, no dose dumping, and a predictable, controlled dissolution profile. In summary, two-stage and multi-stage dissolution testing are powerful tools for evaluating how dosage forms behave across the GI tract. Two-stage tests simulate the stomach to intestine transition. Multi-stage methods mimic complete GI physiology. These approaches are essential for delayed release, modified release, enteric-coated, multiparticulate, and colon-targeted systems. They must follow regulatory expectations for pH, exposure time, and performance criteria. When done correctly, they deliver realistic and predictive dissolution behavior. Thank you for watching. If you found this explanation helpful, please like, share, and subscribe. In the next video, we will cover how to select sampling points and time points in dissolution. And stay tuned because in this around 62 video series, we will dive into dissolution method development, validation, troubleshooting, advanced approaches, and real industry case studies. If you have any question, please post them in comment section. See you in next video. Thanks.